Good morning, afternoon, everyone. My name is Ming Lai, Manager of NGS FAS and NGS Distribution Partner for Rest of Asia at Twist Bioscience. I'm delighted to welcome you all to the Twist webinar, Twist NGS Exome 2.0. In this webinar, you will learn how the new Twist Exome 2.0 maximizes coverage of clinically relevant regions while including updated protein coding gene coverage from key public genetic databases, and how this is amenable to a wide variety of quick and affordable customization options, so you can enrich and sequence exactly what you need. Before introducing our speaker, just a few housekeeping items. All lines will be muted during the webinar. We will answer all questions at the end of the presentation. If you have a question, please submit in the Q&A box found at the bottom of your screen. Following the presentation, if you could please take 1 minute to take a brief survey, we would really appreciate your feedback. And now it gives me great pleasure to introduce today's guest speaker, Dr. Anand Bomakanti. Dr. Bomakanti earned his PhD in molecular and cellular biology from University of Maryland, Baltimore. He has been with Twist Bioscience for the past 2 years as a bioinformatics and NGS field application scientist, helping customers accelerate today's NGS-based research using Twist's range of NGS products. Dr. Bomakanti, please. Thank you.
Thank you, Ming Lai, for the introduction. And good morning, everyone. And thank you all for attending today's talk. I'm Anand Bomakanti, Senior FAS for the COSIA region. Today, I'm really excited to talk about our newest flagship product, Twist Exome 2.0, a gold standard in exome sequencing. So in brief, the value props for the Twist Exome 2.0 is it is one of the thoughtfully curated exomes. It has really very well curated content paired with the best-in-class performance. Overall, it covers 36.5 MB of human protein coding and non-coding genes, and it is based on the latest databases. And it was designed against HG38. And the content has been geared towards being more clinically relevant. Our existing comprehensive exome, as such, one of the best-in-class performance, and we improved over that with improved metrics like uniformity and on-target rate, which I'll be talking about in the coming slides. And along with this, it comes with the exceptional QC process that we have in place for all our NGS products. And it gives you the best data possible that you can rely on. And it also has the flexibility that we offer, where you need not necessarily restrict for the content that we provide. You can always add more content as a spike in to the exome for meeting your research needs, and also at the same time, we have effective. Its performance is effective across multiple multiplexing strategies and target enrichment workflows. Overall, finally, what it leads to is maximized sequencing efficiency, providing the best bang for your buck, irrespective of the sequencer that you're using, and gives you the best data possible for getting the answer that you're looking for. So now taking a little bit deeper dive into the content of our newest exome, Twist Exome 2.0. As I mentioned, it is designed against HG38 database, and the scientists at Twist have taken a thoughtful approach in the content development. Certain key regions and key exons that were either not covered properly or missed are included and ensure that their coverage is like exceptionally good. We included a lot of pharmacogenic genomic SNPs in there, and sample tagging SNPs that are pretty much relevant in the current scenario. And coming to the size, it's a 36.5 MB exome with 43.2 MB design size, making it one of the larger exomes as such, compact, but a little bit larger when it comes to the target to bait probed area. And coming to the performance, you'll see in the subsequent slides, all the metrics are just fantastic. The fold AT is less than 1.35, and then the on-target rate is greater than 90%. And as always, our duplicate rates are like probably best in the class, where we achieve consistently less than 4% duplicate rates. Yeah. And paired along with the exome, we offer multiple configurations of our kits, enzymatic fragmentation kit or a mechanical fragmentation kit, depending upon the type of DNA that you're interested in. And then we also introduced a new hype system, standard hype system with this Exome 2.0 that actually adds to the performance of the current exome version. And also, it provides the flexibility of using our existing fast type. Both give same data-wise. The performance is like same for both the standard hype V2 and the fast type. And it comes with our QC probes. And then the new thing that we introduced is we listened to the voice of the customer, and then we added bead-based protocols to the current exome. So we also supply those dry-down beads along with the current kit configurations, just in case if you want to use the bead-based protocol rather than using the vacuum-based protocol, which has been our standard with the previous exome version. Yeah. So now let's take a bit a deeper look at the content and the design of the Twist Exome 2.0. So undoubtedly, exome enrichment is the workhorse for, exome is the workhorse of the target enrichment, and we sequence around 1% of the protein coding regions and some of the non-coding region during this whole exome sequencing. So what we did is we took a thoughtful approach to get the best coverage possible with minimal off-target effects. So rather than sequencing the entire protein coding region and non-coding region, we kind of put a lot of effort in deciding what needs to be part of the entire design. So again, as I said, we fixated on 36.5 MB of target that is designed around HG38. And then we included all the major databases, GenCode, RefSeq, CCDS, and Ensemble while curating the content. Also, the genes and targets that are covered in the UCSC, because it has its own annotations and RefGene database as well. So along with this, the very important thing we did was looking at the additional evidence to cohesively retain all the exons and target regions that are of importance rather than including everything. So this was a collaborative effort from Twist scientists, field scientists, the bioinformaticians, and voice of the key opinion leaders as such. So what we did was we included all the cosmic pathogenic variants, and then all the genes that are part of the ACMG73 genes. And then we included all the genes that have been identified by the KOLs. And then one of the most important things that we did was we took all the genes that were cited at least 500 times in PubMed, or 500 or more citations for a particular gene. And along with this, we also took any gene that is represented in at least 50 times in the clinical, in the genetic testing registry. So with this particular strategy, what we achieved is we tried to retain entire content of all these genes rather than giving preference to the off-target effects. We try to retain almost all the coding regions for these particular genes. And along with this, we also retain vast majority of the genes that have at least 150 or more citations, or genes that are cited more than 7 times in any test in the genetic testing registry. So we kind of followed similar strategy for the non-coding content as well, where we mainly relied on the ClinVar for determining which regions are functionally important for the non-coding regions. So we tried to retain all the pathogenic variants and likely pathogenic variants, and with minimal effect on the on target rate actually. And then also we tried to retain many ClinVar targets outside these pathogenic variants and likely pathogenic regions. So, for example, some of the ClinVars that are associated with actionable drug targets, metabolism, and also certain variants of uncertain significance, because they actually are part of some of the UTRs and the intronic regions. So we tried to look at all the key genes, we retained all those things, and then also, at the same time, we got target of interest from the KOLs in the field. For example, TERT promoter, right? And I'll show in the subsequent slide. We actually defined the TERT promoter in a much more comprehensive way than the existing exomic content. And then the other thing that we did was we took all the pharmacogenic variants from different databases like PharmaKGB, PharmVar, and CPIC databases. We actually included in this current exome. And then we also included the 2 most popular set of sample tagging SNPs that are currently used. So a lot of effort has gone in deciding what content needs to be part of this exome. And again, to emphasize on that, what we did was we really focused our efforts on key and functionally relevant genes. So for example, if a particular region in a critically relevant gene is not covered because of off-target effects, we try to cover that rather than covering a protein gene with unknown function. We try to cover all the relevant regions that are essential in key genes. That's how we tried to make it a compact exome, but at the same time, its performance and its coverage is no less, or even is actually better than any other content that is present out there. So this is example that I was talking about, the TERT promoter. So you can see that. So the green regions are the twist regions up here, and the magenta, blue, and red are for the competitors. So every competitor defines the TERT promoter in their own way. But here you can see that with the twist exome, we cover the classical TERT promoter region up here that I'm hovering my pointer around. And at the same time, we also cover the enhancer region and some key important regions. So this coverage makes it comprehensive compared to other vendors out there. So it's not just TERT promoter, we took this kind of care with many key genes out there. So the other example is the CFTR gene, and which is one of the key genes that is routinely used in diagnostic settings as such. So you can see that we try to include a lot of intragenic variants as well, along with the standard exonic regions. Like this, for all the key genes, we took enough of care to cover all the key regions that are clinically relevant. So a lot of thought, so you can see how much thought process has gone to curate this content and deliver this flagship product to you. And ultimately, the proof is in the pudding. So let's take a look at the performance metrics of how this of the Twist Exome 2.0. And before that, just a few pointers to bring everyone onto the same page. So essentially, the whole point is to achieve uniformity where every target that we are interested in has the same mean coverage all throughout the exome without any bias, right? This is like the most ideal result that we want to look at, where every base is covered 100% and every target has a mean coverage of 30x. But necessarily this is not the case when we end up getting our sequencing data. And you can see that some regions are at 30x that like we have anticipated, some regions are over sequenced and some regions are under sequenced. And along with this, there are so many regions that have off-target effects, which essentially eat up. So any over-sequenced region or under-sequenced region and the off-target effects essentially eat up the sequencing real estate and adds up to the sequencing cost. So at Twist, we constantly strive to ensure that all these additional sequencing is diverted, redistributed to the mean coverage of the regions that we are interested that you are interested in. And then at Twist, we achieve this by both the design process that we use for designing the panel, also the QC process that we have set in place for during the manufacture of the probes. And the third thing is how we change the workflow so that all these three things add up and ensure to give you the uniformity that you have been looking for once you get your sequencing product. So with the current Exome 2.0, we made, as I said, we made multiple changes to get the best-in-class uniformity. So the main process changes that we did was this entire Twist exome is printed as one single panel rather than multiple panels because of our capability to print huge number of oligos, quality oligos in a rapid way. We were able to print this entire exome in one single run as a non-blended panel. So this actually improves the QC process because it's just one single run. And then we used improved machine learning algorithms for balancing and boosting the probe. So this is one thing that Twist excels in. So we kind of study the regions, understand which regions are captured well, which regions are not captured well, and we try to boost those regions so that the coverage and the capture efficiency is similar all across the target regions that you're interested in. And then the third thing that we actually, the third important thing that we did was change in the actual hybridization workflow. Unlike our previous workflow, we brought about changes in the wash temperatures, and we also introduced new buffer system for our standard hybridization, which kind of improves the performance metrics and also the workflow in general. So as I said, we also introduced a bead-based approach for both the standard workflow system and the fast hype workflow system, so that it gives ease of workflow, and at the same time the performance is of the entire product improves drastically. And finally, again, we have Twist products comes with this awesome NGS-based quality control system where we actually QC all our probes, ensure that the probes are completely represented in the product that is shipped to you. And then you can see here and we have exceptional lot-to-lot reproducibility, so that there is minimal variation between the lots that you receive. And then you have absolutely high confidence on the data that you obtain after using the exome every time. So before going into the metrics, I just want to emphasize on one point that all the metrics that I'm showing to you are generated by third-party lab. These are not our internal metrics. So this all these experiments were done by third-party vendors. So we kind of tried to compare our existing exome with multiple competitors out there. We followed vendor-specific protocols. Apart from vendor K, The input was 50 nanograms for all the other vendors. For the vendor K, we used 100 nanograms. The data was sequenced on a NovaSeq flow cell, and then we generated 6 GB of data for every exome that we have used. All our data was generated on NA12878, one of the standard exomes that is used in the industry. And then we used the current data that I'm showing is with standard hybridization V2 method and the enzymatic fragmentation kit 2.0. One more thing that I forgot to mention is this entire exome is actually kind of tweaked in such a way that it is optimally, it gives optimal performance for NovaSeq. That doesn't necessarily mean that its performance is any different on other Illumina machines, but because as NovaSeq is the preferred production level instrument for exomes these days, we try to tweak the product in such a way that the performance is the best on NovaSeq machine. So here are the metrics that I'm waiting to show to you guys. So first thing is the uniformity. Well, Twist already always has the best uniformity in the class, and we kind of outcompeted ourselves this time. So with Exome 2.0, consistently you get a uniformity less than 1.3. What this means is the bias that is there in the sequences that you get is pretty minimal. And you can see that compared to the other vendors, we are way better when it comes to the uniformity. The next thing is the on-target rate. So this is the area where the biggest improvement is seen in our Exome product. Now we have the best-in-class on-target rate, where we consistently hit greater than 93% with the new Exome, indicating that our coverage is exceptionally well. And also, when you look at the zero target coverages, we are less than 1%, where the other vendors are greater than 1%. And holistically, all these three metrics, the uniformity, zero target coverage, and on-target rate, kind of reflect how well the percentage target bases are covered at, say, 30x. That's what we are showing up here. And compared to the other vendors, we cover 97.3% target basis at 30x with this. So all in all, we have the best performance metrics in the key metrics that we compare when we look at the exome performance: fold-80, on-target rate, and the coverage. So, and most important thing is what this fold-80 means. When in the previous slide up here, I showed that our fold-80 is 1.28, while the other vendors' fold-80 is closer to 1.4, and some vendors greater than 1.5. Is you can see how well all the regions are covered across the AT-rich and GC-rich regions up here. So all you can see that the Twist exome gives a much tighter, narrower, and flatter distribution all across the GC ranges. Whereas if you look at the performance of how these regions perform in other vendors out there, it's just all over the place, right? But with Twist, you can get this really awesome distribution, showing that there is not much bias, irrespective of whether the regions are AT-rich or GC-rich on either end of the spectrum. And usually these AT and GC-rich regions are very difficult to capture. But with the Twist exome, with the new hybridization system, this is even well captured in the Twist exome. And that gives the better uniformity and the deeper coverage where it matters the most. And this performance doesn't change much irrespective of whether you're using the standard hype or the fast hype system. And rather than showing the entire coverage metrics like I did in the previous slide, what I wanted to do is concentrate, focus more on the coding regions. So what we did was we calculated the Picard metrics for the all the coding regions and non-coding regions across the databases. rather than the entire target that is covered in the twist, right? Because exome is more of, we pay more attention to the coding regions. So the idea here is to show how well we cover the various coding regions across the databases like GenCode, CCDS, and for non-coding regions, and coding variants in ClinVar database up here. So you can see, and the other thing is we ensured that this data is 6 GB for every vendor out there, so that there is not much bias in the sizes. And also, as I said previously, we have the largest footprint as such, right? Even though it is 6 GB database, we do have a higher footprint than compared to the other vendors out there. So in simple terms, we outperform all the competitors in every single database category when it comes to the coding regions or non-coding regions. So you can see for GenCode, CCDS, our on-target rate at 30x is like better than any other vendor out there. And so is our performance when it comes to the ClinVar database as well. We outshine all the competitors over there. And then it's not just at 30x, right? Even if you compare this data across various depths, in the sequencing, you can see that at 30x, 40x, and 50x in all the depths, we actually outcompete all the vendors out there. So this is like a very powerful and important slide to show, showing that how well we balanced our target regions and how well we cover them across to the databases, irrespective of the content that is there in those databases. And all this, and again, as I said, the entire all these metrics up here we achieved based, it's a holistic achievement based on the coverage, the design, the workflow, and the QC process that we have in place. And all these things eventually kind of lead to one thing, right? How much are you saving for Twist? As I said, with the competitors, with competitor A, I, and K, you might have to get around 10 GB data, or greater than 7 GB data for other competitors. Whereas with 6 GB of data, you can actually get all the meaningful information that you're looking at from the Twist exome as such. So here you can see that you can save anywhere from $24 to $59 per sample when you're using the Twist Twist Exome 2.0 compared to the other vendors, where the data you need to get more data. That means you have to put with Twist you can put less number of samples onto the sequencer and get the same quality data that will give you the answers that you're looking for. One final thing that I would like to add is the comparison between our existing Twist exome and the Twist exome 2.0. One thing is the new Twist exome 2.0 is a little bit smaller than the current exome that we have, which is the comprehensive exome. Both are amazingly good products, and they have their own value propositions. But again, the Twist 2.0 is actually built on top of the comprehensive exome, where what we try to do is we try to retain all the important genes, add to the content of those genes. Like I said, add more UTRs or clinically relevant SNPs in those particular regions. But at the same time, what we did was we removed some of the genes that are unclassified or have no citations present and stuff, so that the certain content can be removed and new content can be added as such. But at the same time, we are trying to work on bringing out a spike-in panel. So just in case if you're already using Twist Exome 2.0 comprehensive exome, and if you want to switch to Exome 2.0 and don't want to miss out on the regions that are already present in our comprehensive exome, we are working on a spike-in that you can use for ensuring that you get the answers that you're looking for. So in summary, we have come up with one of the best-in-class products, and then which gives really best uniformity out there. And now the on-target rate is better than any other competition. And then it has the most efficient and comprehensive coverage of all the coding sequences from the major databases. And we also try to emphasize and put adding more non-coding region content as well. And then when this product is combined with our silicon-based platform and the NGS QC that you have in place, so it just makes a very gold standard product that will give you really high quality data. And then every time that you use, you can get this high quality data and answer your research questions with confidence. And here are the SKU numbers, just in case if you want to get started on the ordering process. You can always contact our sales team at twistbioscience.com and give it a try for our Twist Exome 2.0. With this, I conclude my presentation, and I'm happy to take any questions that you have. Thank you all.
Thank you, Dr. Bomakanti, for the wonderful presentation. We are now going to dive into some of the questions that have come in. Remember, you can add your questions to Dr. Bomakanti in the Q&A box at the bottom of your screen, and we will try to get through as many as possible. So, for the first question, what is the difference between Exome V1 and V2?
Yeah, let me just... Yeah, so as I mentioned in the talk, the Twist Exome V1 is, the Twist Exome slide. Oh, I'm sorry, I'm just trying to pull out the slide. It kind of disappeared. Yeah, so Twist Exome 2.0 is built on Twist Exome, our Twist Comprehensive Exome. Most of the target genes and targets that are covered in both exomes are almost the same, but we have added more clinically relevant regions from these particular genes. We added more exons, gave better coverage to promoter regions like TERT, which are known to be difficult regions to get covered. And at the same time, what we did was we removed those regions which have no clinical significance or which haven't gotten any citations or known to show higher off-target effects because of the repetitiveness in that. So if you want to know more, just email me if you want to know which were the regions that we removed from current exome, and I'll happy to provide you with the bed file for that.
Thank you. For the next question, does the Exome V2 works as well as core exome panel in cell-free DNA from liquid biopsies?
Yes. So again, as I said, I mentioned the Exome 2.0 is optimized in such a way that you get really better uniformity than our current exome. And when you actually pair this with our enzymatic fragmentation 2.0, which has its own improvements on our existing library preparation kit, it just gives fantastic performance irrespective of whether you're using genomic DNA or cfDNA.
Thank you. Lots of questions coming in. Please remember, if you have a question, please put them in the Q&A box on the bottom of your screen. Next question, what is the definition of the on-target rate?
Yeah, so the on-target rate up here is I don't have a specific slide to show you, but the definition that we showed up here is the exact definition that is used in the Picard matrix, which is the painted region plus padding of around 150 base pairs on either side for the painted region. So that is considered as the on-target region for the metrics that I've shown you.
Thank you. So this is a very interesting question. Any difference in performance and on target for standard versus fast hype?
No. So the performance metrics are exactly the same for both standard hype and fast hype. The only difference that you can encounter when you're using fast hype is a little bit different AT dropout and GC dropout profile, because of the fact that the fast hype was developed for for expediting the entire process. So there is some it's the salt conditions are different and the hybridization conditions are different. So the only place where you can see some differences are in the regions that get dropped out in AT or GC. But apart from that, the on-target rate, duplicates, and the performance metrics are identical. And maybe
Right.
sometimes it's better with fast type.
And I believe we have the protocol for both the standard hype and the fast hype, correct?
Yes. All the updated, the fast type protocol is the same. We haven't changed anything in the fast type protocol. But the new and updated protocols for the standard IV2 are already on our website, or feel free to reach out to me and I can share them ASAP.
Thank you. More questions coming in. Do you optimize 2.0? I assume it's Exome 2.0 on the DNB-seq platforms of NGI.
We haven't really kind of did the experiment yet to see how well how well the metrics will change when you sequence it on a NovaSeq versus a DNB-seq. But I assume that there should not be any difference in the metrics because it was taken into consideration. We did thought about this, but I need to find out how well we have, to what depth we have considered this, the MGI sequencing option with the Exome 2.0. But it is compatible product. You can always use it on an NGI platform, and the initial data that we have shows exceptionally good results as well.
Great, thank you. Next question: what kinds of region Twist Exome 2.0 doesn't cover, although other competitor product cover?
Yeah, so it's kind of difficult to really show all the differences between the competitor product and Twist Exome 2.0, but it's almost we cover 99.9% of everything what the other competitors product cover. So as I said, if a particular gene or region is not there in the 150 citations or 7 genes registry, then probably we might not have given importance to it, especially for the off-target regions. But apart from that, the coverage is very comprehensive when you compare it with the other competitive products out there.
Right. And I believe, if I'm not mistaken, we have the bait files that's available on the website. Am I correct? That
Yes.
You can download and have a look at whether those regions are of interest that you would like to see.
But again, one biggest advantage with Twist is you need not restrict yourself with the content that is there. If you feel that some regions are missing, or if you want additional coverage, please approach us. We can design a spike-in, which will not affect the performance of the base panel, and you will get the coverage of the regions that you are looking for.
Thank you. Next question: how about FFPE samples?
Yes, the Exome 2.0 is pretty much compatible with any FFPE samples out there, and especially with our mechanical fragmentation kit, you can process those FFPE samples and use the Exome for enriching the samples. Yeah, they both are compatible, and yeah, the performance should be pretty good.
Great. Next question. Do you have any data for NextSeq platform?
Yes, we do have the data for the NextSeq platform. Most of the metrics are the same, with minor differences in the duplication rates and stuff. But overall, there is not much difference between the NovaSeq data and NextSeq data. But please do approach me, and I can send you the metrics that we get via NextSeq platform or the NovaSeq platform.
Sure. So next question. Before next question, remind you to, if you have more questions, please feel free to put in the Q&A box. Seems that the questions are still coming in. Next question. How does Exome 2.0 control the percentage of duplicated reads?
So essentially, the entire, it's not just one single thing that restricts the duplication rates. As I mentioned, the processes that are there in the place, for example, how well we do the workflow for library preparation, and then how well we are able to capture the hybridized targets. Means essentially we try to saturate the capture so that entire complexity that is there in the library that is made using the Twist library prep is effectively captured by the Twist probes. Twist. One thing that I haven't mentioned up here, because now it has become industry standard, is we use double-stranded DNA probes. Unlike other vendors who use a single-stranded RNA or a single-stranded DNA probes, our double-stranded DNA probes are very efficient in capturing the entire complexity that is there in the library. So the more complexity you capture, lower the duplication rates. And that's how, that's one of the key ways we achieve these, duplication, achieve very low duplication rates with the Twist Exome or any Twist products in general. Our duplication rates are extremely low.
Good. Next question. If 2.0 has UTR coverage, etc., didn't you find any on-target difference between standard and rapid hybridization? I presume it's standard and fast hype.
Yes. So when I say UTR coverage, the UTR coverage that is provided with Twist Exome is based on the key relevant SNPs or targets that are present in the Twist in the ClinVar database. So that's how the UTR coverage was extended in Twist Exome. So we tried to include every clinically relevant pathogenic and non-pathogenic variant if it falls in the UTR or intronic region. And again, it's just the workflow that we have created that actually ensures that the off-target rates are to the minimal. Rather than that, we didn't do anything special for this, for keeping keeping the off-target rates low between a standard hype and fast hype. It's just the workflow, how well you wash, how well you ensure that the temperatures are maintained. All these teeny things add up to the entire final product that you're looking at.
Sure. Right. So thank you for all your questions. But that unfortunately brings us to the end of today's webinar. I would like to thank all of you for joining us. We hope that you have enjoyed this Twist webinar and would really appreciate your time completing a short feedback form at the about this event. Your feedback is important to us so that we can improve future Twist Bioscience events. Please click on the feedback link that will be appearing on your screen to access the forms. If you'd like to know more information about how Twist products can help you in your research, please visit www.twistbioscience.com. On our website, you will also be able to find the talks from today, as well as recordings of our previous broadcasts. So thank you again, and enjoy the rest of your day.